In this lesson, we'll take a look at the structure and function of the cell membrane. We've already studied the components, their chemistry, their structure, like phospholipids, glycolipids, cholesterol, proteins, etc. So we'll try to put it together in this lesson. So just a couple characteristics of cell membranes. A lot of this we know already. Again, we're just trying to put things together. So you could think of cell membranes as sheet-like structures, and their main job, or one of the main things they do, is they serve as a barrier between the inside of the cell and the environment. We know that it's 2 molecules thick, right? We always call it a phospholipid bilayer. It forms closed boundaries, right? Kind of encloses the cell, as well as the organelles that are membranous. If you remember from our very first module, very first lesson, I'd gone over the membranous and the non-membranous organelles. Well, the membranous organelles contain these same cell membranes. All right, so what we're going to take a look at, kind of put together in this lesson, is that the cell membranes are composed of lipids, proteins. There can be some carbohydrates on the outside, especially of the cell membrane, and we'll see the ratio of the proteins to lipids is going to depend on the cell type. All right, so these are some of the things that we're going to take a look at. Our cell membrane consists of a phospholipid bilayer. Like I said, we know that already, and we know the reason it forms this phospholipid bilayer is that the lipid molecules are amphipathic. Remember that a molecule that's amphipathic has a polar region on one end and a non-polar region on the other end. Right, so the polar region would face any water. Right, so you'll see that there's phosphate heads facing the intracellular fluid, as well as phosphate heads facing the extracellular fluid, while the hydrophobic regions are going to be facing the inside of the cell membrane. Now, the lipid molecules make the membrane mostly hydrophobic, right? We tend to see a lot of these lipid molecules, so as a result, the cell membrane is really going to act as a barrier to things that are non-polar, such as charged or polar molecules. Right? That's what the lipid membrane is really going to act as a barrier to. So we're going to see we have to have a mechanism of moving these charged or polar molecules through the cell membrane, and that's going to be via proteins. Okay, so the main takeaway on this slide is that, again, the phospholipid bilayer is predominantly lipid soluble, so it's going to be hydrophobic. So things that are lipid soluble are going to pass through the cell membrane very easily, while things that are not lipid soluble, things that are charged or polar, are going to have to find a different way to get in. Right, so here's a picture of the phospholipid bilayer. So let's just say this was the inside of the cell over here where my cursor is. Out over here is the extracellular fluid. Right, all of these red circles would be the phosphate heads. They're facing the water out here. The red circles here are the phosphate heads facing the water on the inside, right? Those would be polar, while the green lines are going to be the fatty acids, and those are nonpolar. Now, what holds these cell membranes together? This we haven't discussed yet, so this is kind of interesting. The lipids and the proteins are actually held together by intermolecular interactions. Again, if we go back to our very first module, the very first week or so, we saw that there was a bunch of weak bonds, right? They're typically weaker than covalent bonds, but since there's so many of them, we end up with strength in numbers. This was a concept I talked about our very first week of school. So some of these forces that hold the cell membrane together are the van der Waals forces. They're between the hydrocarbon tails, right? So they're going to be between the fatty acid tails, and then electrostatic, and hydrogen bonding attractions between the polar heads and the water molecules. Okay, so there's three different types of weak forces. Van der Waals forces, they're holding the hydrocarbon tails together, while electrostatic forces and hydrogen bonding, they're going to hold the polar heads together. Um, even like I said, these forces are ultimately weak forces. Since there's so many of them, we have strength in numbers, and that's my last comment over here, guys. The collective aggregate of the weak interactions make the cell membrane a very effective barrier. Right? It does its job well. Now, one of the things we're going to see as we get a little bit further into this lesson is that the cell membranes are fluid-like, right? This is due to that weak, or to those weak intermolecular bonds. One of the other things we're going to see is that the cell membrane is not static, it's not rigid, it's actually like very dynamic, and the lipids and the proteins are in a state of constant motion, right? We'll actually see that the proteins typically move around laterally, right? So that's my second comment. The lipids and the proteins are in a state of lateral motion. We'll take a look at this is kind of like an overview of the proteins, but we'll take a look in detail some of the things that the proteins do for us. They function as transporters. We'll see that some are channels while others are pumps. Some are enzymes. Some proteins act as receptors. Some will mediate energy storage. So we see that the proteins are going to have an important function to the cell membrane. One other thing, cell membranes have polarity, right? Our cell membranes, especially in nervous cells, right, our neurons, create a separation of charges. One of the things you may remember from a regular anatomy and physiology class is that the inside of the cell membrane is negatively charged, while the outside of a cell membrane of a neuron is positively charged. And that separation of charges is actually a form of potential energy, and it allows for the nerve impulse to take place, right? So it turns out all membranes have a little bit of a separation of charges. Where the inside is slightly negative and the outside is slightly positive. Another characteristic of cell membranes is the cell membrane is not symmetric. What I mean by that is the phospholipid bilayer. The phospholipids that are on the extracellular surface side are not identical to the ones that are on the intracellular surface side. So they're going to have two different sets, for the most part, of phospholipids, right? So membranes are not symmetric. This is the next characteristic. The density and the distribution of the proteins is not symmetric. It's going to vary in different membranes. So depending on the cell function will determine these things, right? So the two faces of the membranes differ from one another, right? I'll go over that in more detail in a minute. So those are the characteristics of the cell membrane. Let's take a look at the lipids first. So the way we'll do this is we'll look at the lipids, then we'll take a look at the proteins. So we know that the lipids that make up the cell membrane, we had done this in our previous lessons, consist of phospholipids, glycolipids, and cholesterol, right? So we had gone over their structure again in previous lessons. If you don't remember, you can go back and look at those videos just to refresh your memory. I'll go over some of that stuff again. So let's start with the phospholipids, right? These are the most abundant lipid in the cell membrane, right? So when you typically look at pictures of the cell membrane, all those polar heads with the two tails, those are predominantly phospholipids, right? We had mentioned this before. Phospholipids are amphipathic, that are going to have a polar side as well as a non-polar side, and that's important because this having a polar end and a non-polar end forms the phospholipid bilayer. The hydrophobic tails, guys, always remember, is going to be facing the inside. That's the hydrophobic effect, while the hydrophilic heads are going to be directed towards the water environment, as we've said before. So here's a picture. I actually used this in one of our lessons previously, where we have a phospholipid bilayer, and you can see here these are all the different phospholipids. These would be the polar heads, right? The non-polar fatty acid tails. You can see, one strand is straight. That's kind of showing maybe that it's a saturated fatty acid, while this one is bent. That would be a non-saturated fatty acid. And then over here are some other phospholipids. Okay, so the phospholipid bilayer, again, you can see these phosphate heads are facing the extracellular fluid, while the phosphate heads on this side are facing the intracellular fluid, right? That's the water of the cytoplasm or the cytosol. We'll go back to this picture again. It has a couple good things on it that I like. The second lipid that's part of the cell membrane is the glycolipids, or are the glycolipids, right? Remember from our previous lessons on glycolipids, they're going to contain a carbohydrate. Here, the fatty acid is going to be fatty acids and sphingosine, right? We're going to have a combination of fatty acids and sphingosine. Those are going to be hydrophobic, while the carbohydrate part is going to be hydrophilic. So I took the same picture, I just kind of made it small, and then I took this picture here from our lesson on glycolipids in the previous lesson. Remember that a glycolipid has a fatty acid component. The alcohol was sphingosine. We studied that, and then attached to that is a carbohydrate. We had seen the one where there was like one glucose or one galactose. You can see these are a little bit more comprehensive. So this end of the molecule is hydrophilic, so that's going to be facing the water. The fatty acid tail, the sphingosine, those are hydrophobic. So all I did was I just kind of drew an arrow from here, showing you that would be this over here. Right, so these two tails, 1, 2, kind of represents over here 1, 2, while the carbohydrate is all these sugars up over here. Okay, so these are always going to project on the outside. Notice that the glycolipids are on the extracellular fluid side. We don't see them on the intracellular fluid side. The third lipid that is a component of the cell membrane was the cholesterol, right? This is a compact, rigid, hydrophobic molecule, right? We actually see it right pretty much embedded inside the lipid portion of the cell membrane. It tends to give some stabilization to the cell membrane, especially at temperature extremes. You know, as we go from one extreme to another extreme, or animals live in different environments, the cholesterol helps to stabilize the cell membrane. Oops, sorry about that. Where did we go? It reduces the permeability. It acts like a plug. I'll show you what I mean by that. I'll show you what I mean by that in a second. And it also alters the fluidity of the membrane. So if you take a look here, here's a picture of a phospholipid bilayer. Right. So here's your 1, 2, 3, 4 rings. Right. And remember the OH group? That was the part that was hydrophilic. So you see that hydrophilic part of the cholesterol is going to be facing the water here, while the hydrophobic rings over here are going to be embedded in the phospholipid bilayer. So this here is kind of acting like a plug, so that structures can't seep through the cell membrane this way. So it kind of plugs up the cell membrane. Here's a picture from our book kind of showing the same thing. Again, the circular part here is that OH. Again, if you don't remember the structure of the cholesterol, just go back to the previous lesson, guys, and take a look. That OH part was the hydrophilic part, while the 4 rings, right, your A, B, C, D rings, are going to be your hydrophobic. And that's why we see them embedded inside the cell membrane over here. All right, it's a good picture. So those are the 3 fats. Just a little bit more about these fatty acids. The cell membranes are diverse. Variations in the types of fatty acids has a significant impact on the cell membrane function. Right? The chain length and the saturation play a big role in membrane rigidity. If you remember, we had said that saturated fatty acids, we can kind of pack them together, while unsaturated fatty acids have the bend in it, and we can't pack them as tightly. And that's my comment here. Recall that bent fatty acids, right, the ones that are unsaturated, they're going to pack less densely than the ones that are straight. Also, varying fatty acid content of the sphingolipids influences the membrane function. Okay, so not all membranes are the same. They're going to have a lot of variability. Okay, so one of the things I mentioned, just to kind of finish up with the lipid portion of the cell membrane, is that our membranes are asymmetric, right? The outer surface is always different from the inner surface. So we could look at a lot of different cells. The example I want to give you is just a plain old red blood cell, right? An erythrocyte. Check this out. This is kind of interesting. 80% of the phospholipids of the outer layer are phosphatidylcholine and sphingomyelin. The outer layer also has glycolipids. So remember we said that the glycolipids project from the outside. These glycolipids, they're going to act as a receptor and in recognition functions. But look at the inside. The inside of the inner layer is phosphatidylserine and phosphatidylethanolamine. Right, so you can see the inside phospholipids differ from the outside phospholipids. Right, so this is a great example to show you that the inside and the outside are different, right? And that's for pretty much all of our cells. But this is a specific example of the red blood cell. So again, this is the same picture, guys. I just like this picture here. They're showing us all the phospholipids, right? You can see the red ones are phosphatidylcholine. These kind of yellowish ones, I guess, are phosphatidylinositol or other, or just other ones, while the green ones here are phosphatidylserine. Here, these yellow ones, I'm sorry, these are phosphatidylinositol. So they're showing you that the inside structure is definitely different than the outside structure. And again, remember that the glycolipids only project on the outside. Okay. The last thing we want to look at as far as the structure of the cell membrane are the proteins, right? So besides lipids, biological membranes also contain proteins distributed on the surface, or they can be embedded within the lipid bilayer, right? Or here I put the word integrated. Membrane lipids function as semi-permeable barrier, while proteins carry out most of the other functions of the cell membrane. This is actually an important statement, right? The function of our lipids is to act as a barrier. It's a semi-permeable barrier, but the proteins are really doing a lot of the other work, as we'll see. So just to kind of overview, we had done this in our very first module, the very first week of school, that our membrane proteins, here's some of their functions, right? They can act as transporters. This will be in my next lesson that we'll do. They regulate the movement of substances across the cell membrane. We'll see that there's different channel proteins, there's other carrier proteins, there's protein pumps, right? These are all things we'll look at in a subsequent lesson. Other proteins act as receptors, right? They're there to receive substances that come to the cell. This is a word, I'm not sure we've used it before, but anything that comes to a cell is we can call a ligand. It could be a hormone, it could be an ion, right? So it's just a general term that we use, but there has to be something to receive the ligand. Well, thank goodness we have protein receptors. Protein receptors are bound to the plasma or the intracellular membranes. Right? We'll get more into that in the next lesson. Or they can be found as soluble proteins in the cytoplasm or even in the nucleus. Right? So we have receptors that are either on the cell membrane, they can be on structures inside the cytoplasm, or even on the nucleus. Right? So the receptors are just not on the outside of the cell. Receptor proteins are important in cell signaling and also the intake of large molecules. We'll talk about cell-mediated and receptor-mediated endocytosis in the next lesson, and I'll show you how those receptor molecules are important. Some examples of some receptor proteins that you probably know are insulin receptors, estrogen receptors, there's serotonin receptors, and then even vitamin B12 receptors. And vitamin B12 is going to be taken in by receptor mediated endocytosis. I'll talk about this more in the next lesson. We mentioned it a little bit when we did lipoproteins. This was the way that low density lipoproteins are taken into the cell. So I'll go into this in a little bit more detail in a subsequent lesson. But what I wanted to show you on this slide was just the importance of these different receptors, right? These proteins that are have to be there so that when different hormones or different chemicals come to the cell, those chemicals can work on the cell and have the effect on the cell that it needs to have. Without these proteins, we'd have a problem, right? If we lost our insulin receptors, guess what we have? We have diabetes, right? There's some types of diabetes where there's a receptor problem, right? It's a recognition problem. Some proteins in the cell membrane are identity markers, right? They're used to communicate with the immune system, right? The immune system uses identity markers to distinguish our normal cells from any cells that may have become abnormal or damaged, or even if a foreign cell comes into our body and our immune system doesn't recognize it as the normal markers. And now the immune system can go to work and get rid of it. Other membrane proteins function as enzymes where they catalyze chemical reaction. Others will act as anchoring proteins, which will help to connect cells together, and we can form tissues from that. And then we also have cell adhesion proteins that actually, anchor the cell down to the plasma membrane. Okay, so those are all the functions of the proteins. Again, just to kind of review real quick, transporter proteins, receptor proteins, that's a big category, identity markers, enzymes, anchoring proteins, and cell adhesion proteins. Okay, so how many proteins do we have in a cell membrane? Well, it's not really an easy answer. It's going to kind of range. So the way I did this here is I just kind of put down the mass ratio. We'll do this in terms of mass. The mass ratio of the lipids to proteins, right? How many lipids to proteins is what we're going to compare. So the range can be from 4 to 1 lipids to proteins, where we have 4 to 1, so four times more protein lipids than we do proteins, or it can even be 1 to 4, where we have 1 as the lipids and then 4 times in the proteins. What's going to depend on this is the function of the cell. So for instance, a neuron that's myelinated in the nervous system has a much higher percentage of lipids. That those lipids function to insulate the axon so that the action potential can travel a little bit faster. So a neuron is going to be more on this side, right? 4 times as many lipids to proteins, opposed to, say, the mitochondria, which is going to function in energy production. We don't need as much lipids. We have certain proteins that are necessary for producing ATP. So here we're going to have many more proteins than we do lipids. All right, so those are going to have a higher percentage of protein. So depending on the function of the cell or the function of the organelle, the mass ratio is going to vary. Most cells on general are half and half, right? They're going to be 50% protein by mass. So now that we kind of know the function of the proteins and how many proteins we have, let's take a look at the different types of proteins. The first one we want to mention are known as integral proteins. Now, these are proteins that are embedded within the cell membrane, meaning that they span the entire width of the lipid bilayer. They have a defined orientation with the cell membrane, determined really by their primary structure. So I'm going to take you back to our protein lesson. Remember that the primary structure is the amino acid sequence. So some of the transmembrane segments contain the hydrophobic amino acids. If you go back to one of our earlier lessons, you'll remember that leucine, isoleucine, valine, and phenylalanine as examples are hydrophobic. Right? Those amino acids, they're going to be pretty much in the middle of the protein because they're hydrophobic. So this would be an example. This is a picture from our textbook where we have the phospholipid bilayer. These proteins, letter A and letter B, are good examples of integral proteins. See how they span the entire length of the phospholipid bilayer, right? So those hydrophobic amino acids would be in this general area here of the letter A, or the general area of the letter B is where we could expect to see the those hydrophobic amino acids. Okay, let's make sure we got everything. So these proteins, again, that span the length or the width of the cell membrane, you're going to term them integral proteins. Now, there's different things about these integral proteins, just as a general idea. Your book went into this a little bit. You could read this in more detail. The integral proteins can contain alpha helices. So you remember the alpha helix was the secondary structure where there was a bit of a coil. And one of the examples that our book talks about is a protein that's found in bacteria known as bacteriorhodopsin. And its job is to move protons from the inside of the bacteria to the outside. So here's the picture of it from your book. You can see this whole structure here is the protein, right? This tube right here would have the alpha helix in it. Remember in here that we have like the loops and the turns that we talked about, right? And there'll be another alpha helix here, right? So these alpha helices are acting as a way of protons to move through the protein. All right, so this is something that we see in a bacteria, right? It's called bacteriorhodopsin. Other integral proteins can have beta sheets. If you remember again from our protein discussion, a beta sheet is also a secondary structure. And our book gives another example that acts as a channel. The name of the protein is known as a porin. This allows certain molecules to pass through. So if you were looking at this one, you can see again the phospholipid bilayer, but here you can see the beta sheets. Remember that the beta sheets run like this, and you have some loops and turns, and then this one will be anti-parallel this way, and then this one's going to run this way, right? They run in different directions. You can see all these are anti-parallel beta sheets, but this is going to act as a pore again to allow structures to go through. And again, you can see all these loops and turns right over here. So hopefully this is familiar to you from when we looked at proteins, some of these things that we're kind of putting together now. Okay, so cool, right? The second type of protein, the first one was integral. The second type are known as peripheral. These guys are not embedded inside the cell membrane. They're going to be more on the surface. So these you can think of as surface proteins, which are anchored to the bilayer. Right? The proteins are connected to the surface of the bilayer by different types of anchors. In the lab, actually, these can be easily removed without damaging the bilayer. Or even in the cell, they may have temporary association with cycles of disattachment and reattachment. Right? So these are not as permanent on the cell membrane. They can change. So what do they look like? So we can see here the blue, right? C, D, and E are all examples of peripheral proteins. This letter C peripheral protein here is actually interacting with the polar heads, right? So there's some type of interaction here where this protein can attach to these polar heads. Letter D is interacting with this integral protein, right? So the attachment point is right over here. There's some type of bonding or interaction between this peripheral protein and this peripheral protein and this integral protein right over here. While letter E has a more covalent attachment, right? It actually has a little bit of some type of hydrophobic portion here that's projecting down into the cell membrane. Right? This one's going to be a little bit more securely attached to the cell membrane. But nonetheless, C, D, and E are all good examples of peripheral proteins. Okay, so I had mentioned earlier, now that we know the function of the proteins, the types of proteins, our cell membrane is not a very rigid membrane. these proteins can actually move in the cell membrane, right? So you may have learned this in a bio class or maybe even in your anatomy and physiology class, something known as the fluid mosaic model. The molecules of the membrane are in constant motion, which allows for the proteins to diffuse laterally in this sea of lipids, right? It's almost like you could think of if you take a look at the ocean, up north or way down south, right? They have these big icebergs just kind of floating in the ocean, and it's kind of like that. The protein is the iceberg, and it's just kind of floating through the sea, right? The sea of lipids, though. So the membranes are not static, like I mentioned before. The molecules of the membranes are in constant motion. Now, the fluidity is determined, and we talked about this already, by the proportions of the saturated and the unsaturated fatty acids. Again, remember that when we have more saturated, things are going to be less fluid. When things are unsaturated, they tend to be more fluid. So the unsaturated fatty acid tails, they're big in contributing to the fluidity. And guys, remember the reason for that is the tail of the unsaturated fatty acid is bent. Remember, it's bent at the double bond, and as a result, the tails don't pack together, right? So this fluid mosaic model explains fluidity while the cell membranes are flexible, even self-sealing, right? While how they can be damaged and they can kind of open up and close back up again, right? And we'll see that in our next lesson when I talk about that receptor-mediated endocytosis. And it also explains why sometimes the cell membrane is impermeable. All right, so just to kind of put this back together again, here's a phospholipid where we can see these are probably all saturated fatty acids, right? We can pack them together nicely, right? This portion of the cell membrane would be not flexible. It would be more rigid. While here we have a saturated, but look what's in between is an unsaturated right over here. Yeah, this picture is from your book also. All right, so kind of just putting it together. Now this slide here, we've looked at this a few times, doesn't show the proteins. I wish it showed the proteins, but you can see here again, you can see your phospholipids, glycolipids, some phospholipids, over here different phospholipids, but we could see some saturated fatty acids, unsaturated, right? This whole combination of fatty acids, and you can see all of this contributes to our fluid mosaic model. So that is the structure of the cell membrane. Again, we just took things that we learned from our previous lessons and put them all together. In the next lesson, we'll take a look at how do substances, the different methods that substances move through the cell membrane, and that will be what we'll talk about then.